amazing academic career. If I told you that our next prize winner has an office at the moment that overlooks his high school and is across the road from where he went to university, you might think that he hasn't covered much ground in his life. You could not be more mistaken. He has worked everywhere from Monash, UWA, the CSIRO, to Sheffield University and the University of California, Berkeley, where he is still Professor Emeritus. And as a mathematician myself, it gives me great pleasure to say he's Professor Emeritus in his field of statistics. For those of you who think of statistics purely in terms of dusty book pages covered in bell curves and chi-square distribution tables, think again. Now don't get me wrong, I've got nothing against a good chi-square distribution table. And Bayes' theorem is surely one of humanity's great observations, but this man has moved well beyond. A round of applause for Bayes' theorem. It's the only one I've ever going to do. This man has moved well beyond the textbooks. His ability to interpret statistics and data has seen him work on everything from high-profile courtroom cases around the world to working out the rates of imprisonment for Indigenous Australians, from water use in Perth to his latest work on interpreting the data gleaned from the genomes of cancer tumours. In this age of big data, where geneticists no longer look at a simple single gene, but track thousands of genes in parallel, he's at the forefront of taking the avalanche of information that is presented to him by researchers and helping them understand things in ways they simply never could without him. This is the modern era where mathematicians can gift other academics with insights and discoveries they could never make themselves. And he has done this with aplomb. Ladies and gentlemen, the 2013 Prime Minister's Prize for Science goes to Professor Terry Speed.
I think of myself as a tamer of the line, which is the variability inherent in most numerical processes. As a statistician, I'm trying to deal with variability. Measurements vary when you repeat them, people vary, the internal features of tumours vary. We've got to come to terms with that and see what we can learn despite that variability. It was hard for me not to be interested in medicine. I spent a lot of my childhood in hospital. Well, I started off trying to become a doctor, but when I actually started the medical degree, I realised that I wasn't really cut out to be a normal doctor, you might say. And in some sense, I thought I had given up any medical research plans completely. But over 30 years later, I seem to have come around to it again. Being a statistician, I also wanted to practice statistics. And later on, when I became interested in DNA, my interest and knowledge of biology got me into a reasonably high-profile trial in the US on scientific misconduct, where they were trying to prove using statistics that a certain researcher had fabricated their data. And it was kind of complicated biology, and because I could get my head around it, I got invited to be an expert witness there. In the late 90s in Berkeley and more generally in California, there was an explosion of technological development whereby things that would previously be done one gene at a time tended to be done what's called genome-wide, thousands of genes at a time. And this generates, let's say, 1,000 to 10,000 times more data. So somewhat different techniques are needed to deal with the variability of 50,000 measurements, and the biologists didn't have them. So they needed computational people to help them make sense of their data. I sit in the Robert Isaac Institute. I collaborate with top biologists who study cell death, they study immunology, they study malaria, and all of them produce the type of data that I have the ability to assist with. And so in that sense, I can sort of get closer to understanding disease. The main thing that we do is look at the nature of the genes expressed in cells. Tumour cells, normal cells. How do they change? How does the cell get disrupted to become a tumour? But I'm also slowly drifting towards the clinic rather than the bench, as it were. Rather than the lab stuff, I'm more interested now in what goes on with patients. But of course, they come together. You have a patient, you get some cells from their tumour, you study the cells, you make some predictions. I'm looking at an aspect of thyroid cancer which would remove unnecessary thyroid operations, because in a large category of thyroid cases, out comes the thyroid, and at that point you discover that it wasn't necessary. I'm trying to contribute to the analysis of molecular data about tumours to assist in choosing the right treatment, predicting prognosis, doing better diagnoses, and perhaps ultimately in curing.
In his career, he is slowly drifting towards the clinic on the bench. And as he slowly drifts towards the stage, please congratulate Terry Speed, ladies and gentlemen. A few words, please, Terry.
Didn't bring my glasses, so I can't read it. Well, excuse me, we'll start again. Prime Minister Tony Abbott, distinguished guests, friends, relatives, ladies and gentlemen. I think the first thing I have to say is that I'm enormously honoured and pleased to receive this award, particularly in the year 2013, because I would imagine not too many of you know, but it is the International Year of Statistics. To be perfectly honest, what are we? October? I have yet to go to an event in Australia. I've been to the International Year of Statistics in Mexico, in Germany, in America, but there's one coming up in a couple of weeks in Melbourne. Anyway, I'm a statistician who works, as you heard, on molecular data. It is a fascinating topic. I'm going to get to a little bit of it in a minute. But it's basically statistics and biology sort of linked together. I came to it because when I was an undergraduate, I got entranced with genetics and mathematics. And my hero very quickly became a gentleman named Ronald Fisher, later Sir Ronald Fisher, who was the leading statistician of the 20th century and one of the leading geneticists, and certainly a pioneer population geneticist. He had former students at the University of Melbourne, a professor of statistics, who I was lucky to learn from, Professor Evan Williams, and a professor of genetics, Professor Peter Parsons, who was also one of his students. So early on I was exposed to Ronald Fisher, and as I say, he's been my scientific hero. He came in 1990 to an agricultural research institute in the UK, and he was asked by the director to see if he could get more out of their data. And he created 20th century statistics in so doing. And that's basically been my mantra, or my aim, getting more out of data. Fisher's data, of course, was written on paper, and he did his calculations with machines, you wind the handle. The data I deal with is written on silicon chips. It's basically on DNA, fluorescently labelled DNA. Here is a so-called flow cell, here is a microarray. And these objects, these silicon objects, have millions of pieces of data that you capture by sticking them in laser scanners, reading the images, and then converting it into data that we can analyse. And I'm sure you realise that biologists couldn't do this without help from people like me. So it's been my great fortune, as you heard, to be around at the time when this technology was developed. So that's my main passion. But of course, I'm interested in mathematics, and I'm interested in teaching, and I'm interested also in gender equity. And in some sense, the common theme here is like getting more out of data, getting more out of students, getting more out of women's careers. It won't surprise you to know that many women would like to have childcare facilities. Flexible, affordable, and accessible. We want to work towards that.
Wow.
I have many people to thank for my standing up here, but above all, it's my wife of 49 years and my friend of 50 years, Sally, who's sitting over there. years ago, or 50 Christmases ago, she gave me a present, which was a copy of Statistical Tables by Fisher. That same year, I'm embarrassed to admit now, but she typed my honours thesis about work by Fisher. So Fisher and Sally and I go back 50 years. And she's been a great supporter ever since. I have one brother and 3 sisters, and they and their children and spouses some of them are here with me tonight. I want to thank them because they somehow got me into shape that I could be married to Sally. I had the extraordinary good luck of being made an offer I couldn't refuse in 1997, joined the Walter and Eliza Hall Institute to start bioinformatics. That was by Professor Suzanne Corey, who is over there, and I want to thank her for that. In 2009, she handed over the directorship to Professor Doug Hilton, who's also over there, and he made me another offer I couldn't refuse, to co-chair the Committee on Gender Equity within WEHI. And with his support, we've done amazing things, and we're going to keep trying to do them. All this time, I've needed to be paid, and I've needed the colleagues around me to be supported. So I'd like to acknowledge my debt to the NHMRC, whose Chief Officer is sitting over there. I can't quite see him because I can't see anything, but I know you're there, and I'd like to express my gratitude to the NHMRC. I mean, it probably wouldn't be true to say without them I wouldn't be here, but without them I wouldn't have been earning any money, so to a person like me, it's students and collaborators, everything. And I counted up, I have 8 former or current students or collaborators in the room. I won't name them, but there's a group of 3 that I would like to mention. That's Jenny and Marilyn and Sue, who work together with me and others on the Kangaroo Genome Project, which I think is, roughly speaking, basic research, and we want to thank the ARC for that. And then I have other colleagues and students who have stimulated me, who've made every day at WEHI exciting, and without their stimulus and their interest, really I would have nothing to do. It's great to say he did this and he did that, but of course I don't do it in isolation. What is there left to say? Well, I'm basically a mathematician. I do non-standard mathematics, right? Stuff with DNA. Most people don't know about it. Certainly most school teachers and most kids don't know about it. So I would like to ask you to get the message out. You have children, you have grandchildren, if you're a teacher, you have students. Mathematics is much broader than I think these days than I think people realise. It's not hard to find out how broad mathematics is, but it's not necessarily very widely known. So in closing, I would just ask you to push the students into staying in mathematics, push them into learning about the breadth of mathematics so that we can have a great future together. Thank you.
Sally, if it is true that you really did give Terry a copy of R.A. Fisher's Statistical Tables as a Christmas gift, I am so getting you to speak to my wife at the Terry's going to remain on stage. If the Prime Minister and the Honourable Bob Baldwin would join him for a photo, please, and then we'll bring up our other 4 prize winners as well. So Prime Minister and Bob Baldwin, if you come forward just for a second, we've got a couple of group photos to take before we have a brief break. So please, another round of applause for Terry, the Prime Minister's Once we've taken that photo, if our 4 teachers and researchers would like to also join us on the stage. And please, another round of applause. Sarah, Richard, Angela, and Andrea. Ladies and gentlemen, so much for joining us. We're going to come back in a few minutes and get to meet our prize winners in a bit more detail. It's a short break, and please congratulate them again as they make their way back to their seats. We'll have 5 minute break before some more formalities. That we're hinted at in those speeches and those videos. Please welcome back to the stage Terry Speed, Angela Moles, and Andre Morello. Speed by name, just we've got 20 arts, that's great. Terry, let me start with you. You alluded to this in the video in your speech that you originally considered a medical career. Why and how did that turn out?
Well, I did get alluded to somewhere that I was rather sickly as a child, so I thought, yes, better be a doctor, because I had a lot to do with doctors. Then, as I grew up, it became clear I lacked a bedside manner. So, doing medical research, fine. Started doing combined medicine and science, looked down a microscope, while I seized my eyelashes, tried to dissect a rat that didn't have a pancreas or a liver, something went wrong. I realised that I lacked the kinesthetic skills to complete a medical degree, so I went into mathematics.
How did you fall into mathematics? You know, I should know you prefer the term mathematician to bioinformatician, don't you?
Oh, the bio stuff came much later. You know, start off in pure mathematics, and you sort of get older and want to do something, and then things happen, and people ask you questions, and the rest of the world changes. Bioinformatics didn't exist 20 years ago. The material we studied didn't exist 40 years ago. So you wait till the world changes, and you can go back to the high school, the university, a thoroughly different person.
And one thing, Terry, that people might find interesting, I mean, it makes sense to me what Angela's saying, that when you take on big data and when you look around the world, some of the rules change. But you're suggesting in your video that even in mathematics, when you move to big data, the statistical laws and the practices that you apply change. Some people might assume there's more stuff, but the mathematics would be universal.
Maybe the underlying mathematics is universal, but the details always change. You can have a procedure for doing a calculation that works fine if you have 1,000 samples, or 1,000 units in your sample. But if you have 1,000,000 units in your sample, it may not work very fine. If you have 1,000,000,000,000, it may never finish. So you have to think of different algorithms. So basically, as the data expands, not only do the questions you ask change, but even the procedures that you might want to use, even if they're exactly the same as they would be with the smaller data, you may have to recast them so that they're actually effective with this humongous amount of data. Just reading data into a computer can take hours. So let's find a method. We'll only need to read it once. You do have to read it once if you want to analyse it. Ideally, not more. And that's a challenge. So just things change. I mean, that's research, that's happens to mathematics and statistics just like everything else.
Andre, I'm going to in a second about cancer cells, Terry, but I do have to ask you one question. You worked on the O.J. Simpson trial. What was that like?
Well, it'd be tempting to say it was a circus, and actually it was a little bit, but it was also incredibly stimulating. I think any large criminal trial you see, sounds like a television advertisement, the interplay between justice and truth. Okay, as an expert witness, you're supposed to be there about truth. The whole system is delivering justice according to the justice system, and these things are not all entirely consistent. So, in that way it was very, very stimulating. Evidence, the laws of evidence, how evidence is collected, presented. Yeah, what can I say? It was stimulating. I don't think that I had any effect on it, but it was really interesting to be part of it. And of course, we know what happened.
You're far more at home, drifting between the lab and the bench. What is it your data is trying to find out about cells and tumours? And what is it you can see and you're letting researchers see?
Cancer cells have their genomes messed up in incredibly complicated ways. I mean, it really is still, I think, not quite beyond our imagination, but beyond our ability to... They have mutations, they break, they rejoin. multiple copies. It's just all sorts of aberrations which are obviously part of them being tumours. But which parts are so-called drivers are really important in the tumour process, and which parts are just there because everything is messed up, because important repair genes are not. This is really a complicated task. So somehow, bit by bit, we're getting more and more data. We might get 3 or 4 or 5 or 6 sorts of data on the tumour cells and try to put it together to figure out what is wrong with that cell. Then we have to say, oh, that's just a snapshot. Actually, the tumour from which we extracted those cells, it is heterogeneous inside itself, and it's evolving in time. So one little snapshot, one little piece of one little snapshot, barely scratching the surface of the genomic complexity of this tumour. And that's what we're trying to understand.
Analyse incredible amounts of data. Are we anywhere down the path to understanding complicated systems? I mean, isn't the brain just heaps and heaps and heaps of little things that are happening at one time?
I think this actually boils down to people's personality types. Some people think that we can get to the bottom of complex systems. Others, and I'm one of them, think that it will just get more and more complex as we get deeper and deeper. I don't imagine myself anytime anybody seriously saying, oh, we know about a single cell. Let's move on to organisms or organs. You know, a single cell is the universe.